Okay, so, well, we are going to talk a little bit about the most important aspect of the calcium signaling to the cell nucleus. See, calcium regulates a variety of processes as contraction, locomotion, morphogenesis, growth, differentiation, secretion, regulation of regulation and transcription of some genes. It is not completely established how a single second messenger can coordinate such a diverse effects in a single cell. But there is compelling evidence that it is the spatial pattern of the calcium signaling that coordinate and that specify each one of these signals in the cell. The physiological role of calcium in the nucleus has been known since several decades, but the source of the nuclear calcium continues to be a contentious issue. In spite of the presence of the nuclear pores, results from the various laboratories have demonstrated that nucleoplasmic calcium and cytoplasmic calcium do not equilibrate rapidly. Hence, the concept of a different calcium signaling mechanism in the nucleus other than cytoplasm emerged. The typical resting calcium nucleosol concentration ranges between 100 to 300 nanomolar. Upon stimulation, it can be reached to 350 to 1200 nanomolar. It is also observed that elementary calcium transients produced in the close proximity to the nucleus are transmitted to the nucleoplasm. Calcium puffs generated within 6 micrometer of the nucleus are transmitted almost instantaneously to the nucleoplasm, whereas the calcium puffs generated at 19 micrometer or beyond are not transmitted to the nucleoplasm. Most convincing evidence in favor of a separate nuclear calcium signals came from the electrophysiological studies, which showed that nuclear membrane serves as a barrier for the free diffusion of calcium from the cytosol to the nucleus. Thus, there are evidence for a direct role of calcium in the nucleus distinct from that of the cytosolic calcium. For instance, nuclear and cytoplasmic calcium control transcription of immediate early genes CFOS by distinct mechanism. It is established that the reason that the genes are calcium sensitive is because of a specific DNA sequence motifs present upstream in the promoter region. The cyclic AMP response element and serum response element are the two examples of such sequence motifs conferring calcium sensitivity to gene transcription. Increases in nuclear calcium control transcription mediated by cyclic AMP response element gene, whereas increases in cytosolic calcium activate the transcription mediated by serum response element. In spite of various advances in our understanding of nuclear calcium signaling at the molecular level, there are challenges to be encountered in addressing the source of nuclear calcium. And we are going to look something about it this afternoon. The use of calcium sensitive fluorescent dyes enhanced understanding of cellular calcium signaling, but encountered challenges in the case of nuclear calcium evaluation. This has mainly due to the dye compartmentalization and alteration in dye sensitivity due to binding with different proteins in various organelles differently. Similarly, photoprotein aquarine derivatives as well as calcium sensitive green fluorescent protein derivatives fused with calmodulin camelons gave controversial and conflicting results. Notwithstanding, various controversial results derived from the use of dye, photoprotein aquarine, or calcium sensitive GFP fused camelons. A consensus has emerged from different laboratories that calcium signaling in the nucleus is independent than of the cytoplasmic calcium signals. Now, what is the nucleus? Here, you see, nucleus contains two membranes, the outer membrane and inner membrane. The two membranes are separated by a molecular architecture known as a nuclear pore. So, outer membrane, inner membrane plus the nuclear pore constitute the nuclear envelope. Now, since the question of the nuclear outer membrane being continuous with endoplasmic reticulum, and since we wanted to address the question in an isolated nucleus model, this was imperative that we isolate the nuclear membrane, first the nuclei, then the nuclear membrane, free from any cytoplasmic reticulum or any other cytoplasmic constituents. And this has been achieved through some techniques which have been published and seen in the publications from the Strasbourg group. Next. Here is, you see, this is the work which was done by Joe Paul Humbert, a graduate student in my group, and here is the nucleus. You can see the nuclear membrane with no cytoplasmic contaminants, and at a high molecular level you can see the outer membrane and the nuclear pores also. Now, Joe Paul has further subfractionated these nuclei, and he has obtained the inner nuclear membrane and the outer nuclear membrane. The inner nuclear membrane is a vesicular structure with right side in orientation, which has been shown by the presence of the lamin present on the nuclear lamina on only one side of the membrane, and it is the same nuclear membrane at a bigger modification. Next. Now, this is the first, let me tell you, it was a piece of serendipity that IP3, inositol 1,4,5-trisphosphate, was discovered by Lowell and Mabel Hawking in 1953. The role of IP3 in the nucleus, our role of IP3 in the mobilization of calcium has waited for 30 years. In 1983, Berridge and his associates demonstrated for the first time that IP3 mobilizes calcium from its stored in the cell, which is endoplasmic reticulum. And this followed very hectic activity in the area. For instance, Snyder purified and demonstrated the presence of inositol 1,4,5-trisphosphate receptor on the ER membrane, and Mikoshiba, by molecular cloning, generated 3 species of the IP3 receptor. So, if we are looking of the nuclear event, that means that the first thing to look is IP3 binding activity are present on the nucleus. And this is a work which was published in 1990.